Hey guys, it's Biggs. Welcome back. Now today I'm totally excited about telling you guys about what we're doing today. This is a project that's been on the back burner for a while. I've been wanting to launch it, but we had to launch it the right way. Spend the time, do the research, and bring it to you. So today we are going back to school, and you're coming with me. And what do I mean by that, Biggs? I mean that we are going. We finally have it. We finally have her locked down. She's on board. We are going to be launching a new series dealing with aquatic pathogens, aquatic parasites, aquatic diseases. And it's not Biggs talking about it. It's not some Facebook page. It's not some random YouTuber. We're going direct to the source. We have her. You guys have seen her before. Dr. Denise Petty. She is a North Florida Veterinary Services. She's a well-respected veterinarian in the aquaculture industry, and she's an absolute authority on aquatic animal medicine. She's dedicated her entire life to doing this. So we're going to get into it today, and today we're going to deal with ick. This lady knows her stuff. Everything you've ever wanted to learn about ick, she's going to tell you. Finally have got the legend in the hot seat, and we're going to be going into today. We're going to be talking about, I'm going to let her address that. We're just going to call it ick for short. But Denise, thank you very much for being here with us today. We've been looking forward to this for a while. So I'm going to basically hand it over to you. Take us to school.
Thanks, Chris. And thanks for inviting me to talk about this parasite. It's a parasite that there's a lot of misconceptions, a lot of myths, and I'm just going to lay out some facts. So just a little bit of background to know this parasite, you need to understand how it works, and then we'll discuss how to diagnose it and how to treat it. This probably isn't the most common parasite of fishes, and I know a lot of hobbyists think that it is, but in my experience from looking at thousands and thousands of fishes, that it's not the most common one, but it is the most deadly of the small parasites. It's a very important cause of economic disease. Why? Because it can rapidly spread between resident fish and often has a very high mortality, sometimes 100%. It's thought to have originated in Southeast Asia in cyprinids. There's records of it in back in 1100 in China, where it caused widespread outbreaks. It was first reported in this country in 1898. You have to remember that when the Europeans were moving to the United States, to North America, they brought carp along with them. And of course, that brought along pathogens like ick. And just a note about the name, Ichthyopteris multifilius. The name actually means fish louse with many family members. But ichthys means fish, and so there are a lot of parasites of fishes that start with that ichthys. So there's Ichthyobodo, Ichthyophonus, but Ichthyopteris is the only one. When someone says ick, that's what I think of. I think of Ichthyopteris. The problem with this parasite is that due to fish transportation around the world, and that started long before our time, it's now established in freshwater everywhere. But I want to be sure you understand that does not mean that it's found in every fish tank or every place where fish is. If you're really careful and you're a very careful hobbyist, you can be sure that the fish that you bring in, if they have it, you can eliminate it so you don't introduce it to your fish tank.
So that misconception about ick is in everybody's tank, no matter what, and it's only when fish get stressed that all of a sudden it comes out, that's false.
False. Yes. Thank goodness, because we would, it would be terrible. Life would be terrible if you care about your fish. We would have ick all the time. So what is this? Well, this is a protistan. It's a eukaryote. And, honestly, I don't know if the rest of it's any important. I've got the whole family line there for you to take a look at. The important thing is an obligate parasite of fishes. It must have a fish to live. Now, it can live off the fish, but for a limited time, and I'll go into a lot more detail about that in just a little bit. So what does it do to the fish? So it must feed on the fish. Otherwise, it just will not survive. So it burrows through the outer layer of the skin, which is the epithelium of the fish. And the epithelium covers not only the outside of the skin of the fish, it's on the fin, it's on the eye, it's on the gills. So anything that's exposed to water on that fish has an epithelial layer or epithelium. And whenever the epithelium is damaged, that fish can find it really difficult to osmoregulate, especially the more epithelium is damaged, the harder it is for that fish to maintain its electrolytes within the body. You have to think about a freshwater fish is like a little plastic bag of water that contains a lot of electrolytes, chloride, sodium, potassium, calcium, magnesium, and more, but those are the basic ones that are critical. And they're just like us. They have to have these electrolytes, have to be in the right balance to live. And if anything messes it up, like if you poke a hole, let's say we put our bag of electrolyte water into a freshwater tank, and then we poke a hole in it, what's going to happen? Well, water from that tank will go to where it's saltier, and the inside of that freshwater fish is a lot saltier than its environment. And I know this is probably a little bit of basic, but just to understand what's happening with this fish, when it is burrowing into the epithelium, it's making a hole. And it's not just one organism, it's usually hundreds, perhaps thousands of them, making holes in the epithelium of the fish. And this fish will have a very difficult time to survive. So not only is the parasite feeding on it, this fish is suffering from osmoregulatory distress. The other thing is, if the fish, even if the fish does survive the egg outbreak, it can die from secondary bacterial infection, because those holes in the epithelium can allow bacteria that normally wouldn't be a problem, but now they can easily come in. It's just like opening a door into the fish. Yeah. Let's talk a little bit. I'm sorry, did you have a question?
Nope, not at all.
Talk a little bit about the shape of the fish. And actually the photo up on the upper right that you can see, that is ick as viewed under a microscope. It's live. That's what we call a wet mount, where you take a prep from external tissues, put a drop of water from the aquarium, and then cover it with a cover slip and take a look at it. The yellow backgrounds, because I was using like, excuse me? Oh, yes. The yellow background is a farm microscope. So it's usually spherical, and it's holotrichous, which means it's covered in cilia, and all life stages are motile. So even when it's under the skin of the fish and feeding, it's still motile, and it's moving around. So you can imagine if you had a pretty large parasite underneath the top layer of your skin moving around, you'd be pretty itchy and you want to scratch it. And the fishes do the same thing that have this. The size is variable depending on the life stage and the length of time it feeds on the fish. So the longer it stays on that fish feeding, this organism just gets larger and larger. And you can see in that photo that light area that looks like a little smile, that is the macronucleus. They have a macronucleus and a micronucleus, and it's not important except that horseshoe-shaped or C-shaped macronucleus is distinctive. That is only found in Ichthyopterius. So if you've got you can just almost use a hand lens if you've got a good hand lens, because you can easily see this view of it with a 4X lens and make a diagnosis. Now the theronts are much smaller. They're teardrop shape or oval shape. They're also covered in cilia, just like the adult, I hate to say mature stage is. And all of these they move pretty slowly. And this is the life cycle. It's important to understand the life cycle, because this is how we can eliminate it and try to keep it from killing more fish. The trophont is the large stage that we see in the photo on the upper right. And that is the feeding stage, the active stage feeding on the fish, where it's underneath the epithelial layer, which is why I don't care what you put in the water, you can't kill it unless something you put enough to kill the fish, and then the parasite dies. And obviously that's not our goal. Our goal is to kill the parasite and keep our fish alive. But you can't get to it. I don't care what anybody says, you cannot get to it when it's feeding on the fish. We have a limited time when we can kill it once it comes off. So if you look here, we have it here on the fish. When it is mature enough, it leaves the fish, and usually it attaches to substrate or the tank glass or something inside the tank, and it secretes a thick cyst wall around it. Now, when it's swimming to become this stage, which is called the tomont, it takes it, a lot of this is dependent on water temperature, which we'll talk about in a minute, but like at tropical fish temperatures, it's only going to take an hour or less for that. So you've got a very limited window, and you can kill that stage when it's coming off the fish. Once it secretes that cyst wall, it cannot be killed. It's impervious to any chemical that we have, short of, again, killing the fish.
You mentioned earlier, Denise, about that, like we've all seen it, where fish will scratch against things in the aquarium. Can these be dislodged? Like if you have an African cichlid scraping its side against rocks, can it dislodge some of the adults?
Well, if it can dislodge it, that means it just scraped off that epithelial layer.
Made it even worse. Yes.
Yes. So now you have another problem.
True.
But yeah, but obviously the fishes are itchy, and I'm going to talk that behavior is called flashing. Especially silverfish, that they go and scratch, and when they twist their body and do it, you can see a flash in the water. And that's, they don't have hands, so that's, they can't turn around and chew at themselves like a dog or a cat would do to their skin. So that's the only way they can. But I think it's pretty hard. I mean, if you do a skin scraping, you usually can't even dislodge them if you're dragging a cover slip gently across the surface. You'd almost have to really press hard. And again, you'd be taking off the upper layer of skin when you do that. So I don't advise doing that.
Well, I think about, me with poison ivy. I'm so really allergic to poison ivy. When you do finally get it, it's impossible not to scratch it. And every time you touch it, it spreads.
Exactly. Yes, you're spreading that every time. Well, the fish, it doesn't work quite the same, but they probably do have that same sensation that we would from either like a mosquito bite or a fire ant bite, or like you said, with poison ivy or poison oak. So inside this tomont divides and become with daughter cells, which are called tomites. The problem here is that one trophont can produce hundreds to thousands of tomites. So it only takes one trophont getting into your tank, and now you have a problem. When the cyst will rupture once those tomites mature, and when they swim out, now they're therons, and they swim through the water, hoping to bounce into a host. And again, temperature plays a role in this, but at tropical fish temperatures, those therons can live for about 4 hours, swimming in like a spiral fashion in hopes that they don't hope, but if they bump into a fish, they can burrow through the epithelium within 5 minutes. So again, they're not just sitting there. I mean, they're made to get into that fish quickly so they can start working on the next part of their life stage.
And this is a healthy fish, correct?
Any of them. Yeah,
so they don't necessarily have to be a sick or stressed fish or a fish that has an open wound. This is purely totally healthy fish. This can happen.
That is correct. Yeah. Perfectly healthy fish. This parasite, I mean, this is what it does. It lives off of fish. It has to have them. So it's obviously evolved to use a lot of different strategies to its best so it can ensure its survival. So this theron stage, this is our treatment goal. When we put a chemical into the tank, this is what we're trying to kill. And this is going to be important. So you think about this life cycle. If you have a case with ick, and you're going to understand why I'm going to talk to you later about treatment. Let's talk a little bit about the effect of water temperature on Ichthyopteris. So this table comes from a really nice 2-volume set. If you can find it, I'm glad I got this, but it's awesome. And we're looking at tropical fish temperatures here on the left. I don't know, I've got both Celsius and Fahrenheit, depending on what you're most comfortable with. But if you look at typical tropical fish temperatures, the life cycle in days is 3 to 4. There's some caveats with this, and we'll discuss those in a moment. But as the column, if you go down the column, the water temperature decreases, and you can see that the life cycle increases. So ick is incredible. It's a parasite of all kinds of freshwater fishes, those found in cold water, those found in tropical waters, those found in temperate waters, all freshwater, of course. And even they can tolerate a little bit of salinity. Now, here's where the problem is, because I know that raising temperature is often mentioned as a way to control it. Unfortunately, ick is there's a lot of different types of Ichthyopteris multifilius. There's different serotypes or isolates that produce different antigens, and I'm just going to call those variants, or you may hear me say strains. And just to give you an example, some researchers back in 2006 used 2 different serotypes to infect fish. So they ran a trial where they had the same environmental parameters. The water was similar pH, similar water quality, similar temperature, similar filtration setup, and they used the same species of fish. So they tried to make it as similar as you possibly can, and of course had a control group and replicates. One of the serotypes had a much shorter life cycle, about 7 days, but it caused 100% mortality rate. So this was a very virulent strain in that it took a short time, but went so rapidly that none of the fish survived. Whereas the other serotype had a longer life cycle, but only resulted in about a 50% mortality rate, so but up to 10 days. Now, I don't really know, trying to find papers on all of the different serotypes, I've just found a couple, so I can't tell you where they are. But this means that your treatments is going to have to keep that in mind. You can't use a short treatment period, like I know the label on some of the aquarium store medications say treat 3 times. That may not work. That you may end up having a treatment failure. Why? Because maybe the strain that you have is one of the ones that takes a longer time, or you may have one of the ones that's very virulent. And if you don't recognize you have a problem, identify it rapidly, and get the treatment going, the proper treatment going, then you may lose all your fish no matter what.
And this is the one.
Those situations.
This would be the one that people refer to often in the trade as like the new strains, the super icks. And like anytime anybody posts about ick, they say you got to do this, this, and this. And then all the other people will jump on saying, oh no, it won't work anymore. It doesn't work for ick anymore. So it's not the fact that it's one. You're mentioning there's many different variants of it now, and it's always evolving.
Yes.
Okay.
Right. So yeah, if you had the strain that's very virulent and you didn't react quickly enough, the fish are probably all going to die. It doesn't mean you shouldn't go ahead and try to treat. Right. But yes, or if you've got one that has the slower one and you only treat for say 3 days, it's not done. It's still working. So it's still, you may look and the fish may look better because you help drop the numbers in the tank. And we're going to talk about immune status in just a minute too, because then, you kind of help the fish survive. That doesn't mean that the parasite is eliminated. And that's going to be their goal, is elimination. Now, there was a paper from 2001 where a couple of researchers reported a pretty severe outbreak of disease in some fish in Southeast Asia. They said it was a new species of ick, Neo-Ichthyopteris schlottfeldtii. I think I'm pronouncing that correctly. And it was supposed to tolerate much warmer temperatures than Ichthyopteris multifilius. And it differed a little bit by having a spiral-shaped micronucleus, and it could reproduce on the fish host, which means, I just talked about the stages that were off the fish. I have never seen this one. So I don't know. And I've looked at, many, many, many fishes. Doesn't mean.
That sounds scary, because if it reproduces on the actual host, your method of treatments are often the ones that we're used to are thrown out the window.
Yeah. For me, the jury's still kind of out on that one. I guess when, some of my colleagues or I see that one, I'll feel a lot better about it, because I have colleagues that, they look at lots of fishes every day, and this is something that would be distinctive enough that you would notice it. But I throw it out there because there is that report, and it has been in a couple of hobby fish reports. So just in case. So one of the things that can influence susceptibility, which means what makes the fish more susceptible, it's believed that all freshwater fishes can be infected because there are reports from numerous species of freshwater fishes from all around the world. But you may notice that sometimes you have some fish that have it, and other fish in your tank don't appear to have it. Now they may, if you're not doing gill biopsies and fin biopsies, if you're just relying on grossly looking at that animal and looking for white spots, that's not good enough. You need to, take gill samples, and that can be done so that it doesn't kill the fish, but that's another story. But if you're not looking at gill tissues and fin tissues and skin scrapings, you can't say that fish doesn't have it. But it's possible that there's genetic differences even in the same species, the physiological status, which you just kind of hinted at. Maybe this fish is a little bit healthier for whatever reason, because there could be other underlying disease in animals that you don't even know about. Environmental conditions, nutritional status, mucus production, and there's many others. So there's a lot of variabilities that can influence that.
So
and what's really
posting a blurry picture on, say, social media is not the most definite source for disease identification, is what you're saying.
And honestly, even if it was a clear-cut picture, I'd want to have the microscope, and let's do some wet mounts, because, yeah. there's what you think is ick sometimes isn't, but I'm going to cover that in a few minutes. What's really important for hobbyists is that survivors, if you have, say, 20% of your fish survive, doesn't matter, the survivors of ick outbreaks often will have a very short-lived immunity. And I've looked at some of these fish. So, and to you, if you're not, again, doing those external tissue biopsies I talk about, you don't see the white spots. So you go, oh, this fish is good. I killed ick, it's gone. Doesn't mean that it's gone. There are ways that we can try to assure that we're going to get rid of it. And I'll tell you how to do that too. But I have looked at fish that have been through an ick outbreak and done skin, fin, and gill biopsies on a fish that grossly appears perfectly normal and looks healthy, and seen a couple of trophonts on the gills. So yes, maybe I didn't see anything on the skin mucus, I didn't see anything on fin biopsy, but it's still there. And it's still, which means if you're seeing trophonts, that means it's still reproducing. It's just at a low level. And when those little theronts are coming out, the fish may be able to mount an immune response to keep the numbers down so it's not severely impacted by the parasite. But it doesn't mean that the parasite isn't going away. And if something changes, if some stressful situation occurs, or you buy new fish, you don't quarantine them and you put them in, that, those fish will definitely be stressed. Transport, handling, things like that are huge stress inducers in fish. Those fish can re-break, and then you have an ick outbreak all over again. So a little bit, some of the research has looked at the environment on ick, and by environment, I mean the water that it's living in. The temperature range for it has been reported to be anywhere from like 1 to 30 C, which is, that's just above freezing. It's amazing, to up to some pretty warm water levels. Tropical fish can't, actually we have some temperate fish here in Florida that have to deal with 90-degree water temperatures in the summertime. But there are some variants that could easily survive at 91 degrees Fahrenheit. That's pretty doggone warm. And I would not subject a sick fish to that kind of temperature and expect them to survive. So that's just, I'm putting that out there. Let's look at the salt level. I love salt. If you have a fish that will tolerate salt, it's awesome. But the research has shown that salinity is above 5 grams per liter, which is the same thing as parts per thousand, totally inhibited development. But that was one study. But even at 5 grams per liter, the developmental time increased and the number of theronts decreased. So we see that the salt did somewhat affect the life cycle for that particular variant that was being looked at in the research study. So keep that in mind. And then another one is pH. Well, the tomate stage, which is that cysted stage that is on substrate, it survived a pH of 5 to 10, which is pretty remarkable. You don't see that many freshwater fish species living at a pH of 10. And the theront stage survived a range of pH from 4 to 10 for 1 hour. So, tough as nails. They're really hardy. And I'll put the links for the papers if you want to look at those.
Now, well, you mentioned 91 degrees because the standard treatment before was what? It cannot survive past, what was it, 86.4 degrees Fahrenheit?
Right. Well, and of course, that was based on that the temperature range reported to be 33.8 to 86. So, yeah. Obviously, things change and some can survive. So, you have any questions for me?
No, you're way too thorough. You're wonderful.
Then let's move to diagnosis. How are we going to make sure of what we have is ick? So first we can look at clinical signs. Unfortunately, they're not specific. Lots of sick fishes will exhibit these clinical signs, but you might see clamp fins, dark coloration, rapid respiration, especially because if it's on their gills, it's impairing their ability to get oxygen, which is obviously a big problem. The flashing behavior we've discussed, when I've seen them, usually they're hovering near the tank bottom, but that doesn't mean that's a given. Often, because of the impact on their gills, they may gather near an aeration device, or if you have incoming aerated water, they're going to go there where the oxygen may be higher to try to get more oxygen in. And the mortality curve typically is the first day, there may only be a few fish, but the next day there may be 15 fish. The next day there may be 25 to 30 fish. And the next day it would be 100. That's been my experience when I'm working with large numbers of fish, and it looks like a few fish, and all of a sudden, I mean, within a couple of days, you're losing, 50% of the population. And the white spots that are classic, they may be visible on the body and the fins. But that doesn't necessarily mean anything. For me,
sorry, I lost my train of thought for a second.
Right.
When you mentioned the gill arches. The gill arches are often a case where nobody would see the adults on the gill membranes or anything like that, because that would all be covered by the operculum. But is that
right.
the primary attack zone, because the gill arches and the membranes are a softer tissue, easier to penetrate than, say, the exterior and the scales?
Well, now you have to remember, they're not actually going through the scale. They're going through a layer of tissue just on top of the scales.
Yeah, the epithelial layer.
Yeah, and it's the same on gills and inside the mouth, the branchial cavity where the gills are. And people don't think about that, but that, it will actually go in, and in fact, on the inside of the operculum, all of that's covered in epithelium. And if you do histopathology, you get to see that kind of stuff fairly frequently. But I'm not sure. I've seen it very often on gills, but I don't know if anybody's really done a study looking at it. I think that they're probably just whatever it bumps into. If it's a fish, it goes in. But it's a good question, and I don't know the answer. I don't know if anyone really knows the answer. So I've already mentioned this, but doing external tissue biopsies, skin mucus, fin, and gill biopsies, those are critical. And I always do all three areas, because so many times I'm doing a necropsy, or I've been working on a live fish, and I don't see it on either fin or skin, but I find it on gill. Sometimes I find it on a fin biopsy, but I don't find it on gill or skin biopsy. So if you're only looking at one place, you may miss it. But the problem for trying to make a diagnosis off of a photo or just looking at that fish in your tank is there are quite a few parasites that can have a similar gross presentation. Mm-hmm. And if you don't do those biopsies for microscopic examination, you may misdiagnose them and treat the fish like it has something that it doesn't have.
So an average home aquarist, if they were to go on Amazon or a scientific supply and buy a very basic student kind of microscope, what magnification would they need to be able to see that macronucleus, to definitely know that's what they're dealing with?
Easily with 4X.
Oh,
at least 4X and 10X. Yeah,
readily available to every single hobbyist should they want it. Yes.
Yeah, and most of them are going to come with a 4X, 10X, 40X. So that's magnified 40 times, 100 times, and 400 times. You do not need the 40X to look at ick. And if you want to look at bacteria or a parasite like Ichthyobodo because it's so small, yes. But for ick, it's a huge parasite. You don't really need to go up that high. It's easily diagnosed with that 4X objective. Just one thing I wanted to caution you, and especially this is important if you buy new fishes and you have them in a quarantine tank. If you go just off looking at them and thinking, well, they have ick, those fishes, it's not uncommon for those fishes to have other parasites. And I know that because I've looked at so many. And then at least you'll know that they have them. Because when you're done, the first thing I would do is always treat it first. That's an emergency parasite, in my opinion. You treat it, eliminate it, then you focus on treatment for those other parasites. Depending on what they are, one, they may not respond to the treatment that we use for ick. Two, they may not even be treatable.
Right.
And the nice thing about making that diagnosis by using that microscope, then you can make a decision on that. If you don't, you're just guessing. You're using the best guesstimate.
And I think that's part and parcel why today in North America it's become real prevalent that all of a sudden all of our medications are no longer available, because people as aquarists are just randomly using products willy-nilly without any actual science behind it. They're just guessing it by a picture.
Right. Yes, well, you can look at those medications in the pet store. What does your fish have? What does it show? Oh, it has this. Oh, then here, you use this medication. That's terrible. Just makes me shiver to think about it.
Cartoon drawings, that's all you need.
Yes. It's a scary situation. That kind of therapy is what I call shotgun therapy. You just pull everything out, load both barrels, and just start blasting and hope that you kill it all. Not the best option to do.
I've seen it at some of the farms where they literally, I view it as nuking. After they take just literally bomb the entire thing, turn it neon green or something. It's like, hopefully everything that was there is killed.
Yes. And then of course, some of these chemicals that are available aren't that effective. And I believe in using the most effective chemical possible that also has, hopefully has the best safety for the fish.
Right.
A lot of things that we use are toxic to everything, and it's all dose dependent. But I'll get to that a little bit later. So first, I want to show you a couple of examples, gross examples. These aren't the best photos, but I'm usually making microscopic photos. But this is a red-headed bricon, and you can see these little bitty bubbles here. Well, I couldn't see white spots on this fish. I mean, there's a white spot, but I don't know that that's not a little bit of a guanine pigments in the scale, that silvery pigment that can sometimes, look white at a glance. And then this is a pike cichlid, and it's covered. You can easily see white spots on the head, on this pectoral fin here. And if we went further back, we would see them on the whole body. Does that fish, there's even one on the eye here. And of course, it can do that too. So any fish that has eye issues, you always look carefully because there may be something on the eye. Can I say which fish has it? Only because I did the biopsies to prove it. And here's the example. So this is a skin scraping viewed through a microscope. This is using that 4X objective. You can just see just how large, and we've got different sizes here. This is a mature trophont. The larger it is, that means the more it's been feeding on the fish. These are smaller, so these obviously are a different age than this one. And then we have two small ones that kind of look like fat teardrops. Those are theronts. So I was actually able to catch two stages. So these haven't even been able to, well, maybe they were just boring through the skin. And when I did my skin scraping, I picked them up. Now here's a scale. A lot of times fishes that have a disease or like ick or some of the other parasites, the scales will get loose in their scale pockets. They're in the skin embedded. And normally they're pretty firmly adhered, but when you have any kind of inflammatory process due to a parasite or a bacterial disease, the scales can get loose, and sometimes just dragging a cover slip over will pop a scale out. So that's what this clearish thing we can see these lines in. That's a scale that came out with it. Not ideal because now that means there's a really big hole there. But so yes, this fish did have ick. Now here's a fin biopsy from this pike cichlid, and you can see it looks completely different. Yeah. And this is an encysted parasite, but this is a Digenian trematode. So it's a stage of the Digenian where it's encysted and these guys, there's hundreds and hundreds of them, and each one has a preferred site. And this one goes underneath the epithelium, and it's waiting for that fish to be eaten, probably by a bird or a larger fish. But so right away, I would not waste my time in treating this fish for ick because it didn't have ick. Now I also looked at gill and skin, and it did not have ick. So, but this one does. So this would be like emergency, start treatment immediately.
And that pike cichlid, that has all the classic traits visually of what people would associate with ick.
Exactly. And you don't know how many times I have looked at a fish and I go, oh, I think that fish has ick. And then I come to find out, wow, it's got something different. It's often sometimes something really strange. And so I was wrong. And sometimes I look at a fish and I can't see anything that would even suggest ick, but yet I find it on my biopsies. So at this point, in as many fishes as I've looked at, I never say that fish has ick by just looking at it.
Yeah.
I always want to look at tissues under the microscope, and then I can tell you whether the fish has ick or not. Now here are some more wet mounts. This is a lot of mucus. Often these fish respond when they have ick with tons of copious mucus. And that they do that with almost any external parasite, actually. Ichthyobodo causes the same response. Anything that's really irritating, the fish will produce a lot of mucus. And the mucus does contain compounds that have some cidal activity. So, there's a reason why they're doing this. But you can see there's tons of different sizes of trophonts within this mucus here. And this is just one that's swimming off the fish. Unfortunately here, we can't see the cilia, but well, actually I can see here, but I've got a couple of videos that you'll be able to see the cilia constantly moving. And you have to remember, this is a still image, but if we were looking at it in the microscope, it would constantly be rolling. And so this macronucleus would be rolling around too, as this animal organism is moving around. And inside are vesicles, they're organelles, food vesicles, the basically machinery that helps keep the organism alive. Now this is an example. This is a gill biopsy. I can actually tell because of that right there, probably from a catfish. And this is Piscinoidinium. But what Piscinoidinium, this stage will never move. It's kind of a fat teardrop shape, and it does not have that macronucleus that we can clearly see here. We can see kind of it here. Anyway, if you're looking carefully, you can see the difference. So there's lots of things that look similar, but pay attention. Is it moving? Can you see cilia? Can you see the macronucleus? Now, these are wet mounts of gill biopsies. If you've never looked at the gills under the microscope, this is what healthy, normal gills should look like. And I had to make myself make a photo one time because I tend to only make photos when I see something that's bad. But, anyway, just so you can see, this is what a normal gill should look like. So here's a primary gill filament. Here's a large trophont. You can see how damaging this will be just from the size of that big trophont on the size of the gill. Here's another one. And remember, and you can see how this is the outer layer of epithelium around that gill, and it's bubbled out because the parasite is underneath it. And it's constantly moving and rolling around inside. Here's another gill biopsy with lots of trophonts. This was a small fish. This is a whole gill arch. We can see the cartilage arch here with the primary gill filaments coming off. Here's a big trophont. You can see that beautiful horseshoe-shaped nucleus in the trophont here. There's another, probably a very young trophont here. So I'm hoping you get the idea of just how bad this thing is when it's on the fish.
Seeing them on this scale, when you blow them up like that, it's very, very easy to see for anybody, even a basic aquarist, to see how incredibly irritable this would be to an animal. Imagine something, this is the equivalent of taking like a golf ball and putting it underneath your skin and making that golf ball move or vibrate constantly. That would be so irritating.
Exactly. Yes, it's actually horrifying to think about, I think. Thank goodness we don't have parasites like this. It would be terrible. And I want to show you one last example. Again, this is a gill biopsy. We've got a trophont here, a trophont here. This is a different parasite. This happens to be an encysted Digenian trematode. So it's got a little cyst wall. It's actually in the cartilage of that primary lamellae. So this is an example of another parasite. But honestly, I've looked at a lot of fish that not only did I see trophonts, I saw Ichthyobodo out here. I saw monogenians, what a lot of hobbyists call flukes. I mean, you got just a whole grab bag. What parasite does this fish have? And it has maybe 5 to 10. Yeah. So, take a look. And if you're going to do this, look for other things too. So they're there, because when your fish if they're still having problems after you've properly treated for ick, there was something else that now has kind of taken over and the fish just can't cope with it. Of course, there is no treatment for this one. So, but at least you know it's there, and it would be in stage anyway. And just to show you one last wet mount, you can see, look at the epithelium on this gill here. You can see how it's a little bubbly. It should be a thin layer, and over here it's really thickened, and look at how elevated it is off the gill. You can see this is where there were trophonts, and they've left that fish. And over here is an area that's undermined, and this is typical. There could have been 3 or 4 trophonts living right in this area, just and taking the epithelium off. So if you think these fish don't have gill damage, yes, they do. And just real quickly, and because you probably would never look at this, but this is histopathology, where I preserve some tissues from a fish in formalin, 10% neutrophil for formalin, and then had them processed. And I wasn't looking for ick. I was actually looking for something else. But this photo here, you can see my mouse cursor, can't you?
Yes.
Okay. This is a cross section of skin. And this purple layer is the epithelium, the top layer of the skin. And normally it should be at about this layer. And you can see how ballooned it is. So, oh, there goes my dog. This is ick here. This is a muscle tissue underlying it. And there's these tiny little blue dots here are inflammatory cells, white blood cells coming in to try to help fight the infection. This is a fin biopsy. This is a cross section of the fin. This is cartilage. This bright pink spot is cartilage. Here's epithelium on this side, epithelium on the other side, and this is ick. And I think there's one maybe in here, but you can see the response that the fish has. This is a pretty normal gill, the secondary lamellae on a primary gill filament. This is a gill arch here. You can see, this is how they're supposed to be. They're like the radiator of your car. The gills are designed for surface area to extract as much oxygen from the water as possible. Now here's a fish here, and you can see that we don't see these nice little fingers sticking out. Yeah. Why? Because there's ick. We've got ick here, ick there, ick there. We can see some fat fingers here, but so these gills are toast. Oh, and here's the gill arch. There's an ick there. So I'm just trying to drive home how nasty what this thing is doing to the fish. Don't take it lightly.
Now, wasn't that scary? Now, next video, because this video was a little bit longer, we had to split it up. So you guys learned everything you never wanted to know about this parasite. Ooh, scary stuff. But the next video follow will be all the preventative and how to deal with it and how to treat it. So stay tuned. Till next time, guys. Take care.